Our journey now arrives at another remarkable organelle. Ahead of us, a series of smooth, flattened membrane sacs forms the Golgi Apparatus, the cell's central processing and distribution center. A transport vesicle approaches the receiving side of the Golgi. This entry point is called the cis phase, where proteins arriving from the rough endoplasmic reticulum begin the next stage of their journey. The vesicle gently fuses with the Golgi membrane. Its protein cargo is released into the first Golgi compartment, ready to move through a sequence of specialized processing regions. As we move inside, newly synthesized proteins come into view. Although their basic structure is complete, they still require important finishing touches before becoming fully functional. Moving closer reveals the intricate three-dimensional shapes of these proteins. Their precise structure determines how they will function once they reach their destination. Surrounding these proteins are the stacked Golgi cisternae. Each compartment contains a different set of enzymes that modifies proteins step by step. As proteins travel through these compartments, they encounter specialized enzymes. Each stage carefully prepares the molecules for their specific role within the cell. One of the most important changes begins here. Small chemical groups are added to proteins, altering their properties and preparing them for accurate cellular targeting. Notice the branching sugar chains being attached. This process, called glycosylation, helps proteins fold correctly, remain stable, and carry molecular identity tags. Additional sugar residues are carefully trimmed or extended. Even small changes in these carbohydrate chains can influence where a protein will ultimately function. By now, many proteins have completed their modifications. Each one carries its own molecular signature, allowing the cell to distinguish one destination from another. The journey now reaches the Golgi's sorting region. Here, thousands of different molecules are organized, ensuring every protein is directed to the correct cellular location. Proteins with similar destinations begin gathering together. The Golgi efficiently separates cargo that will remain inside the cell from cargo destined for secretion. The membrane starts to curve outward around the selected proteins. A new transport vesicle gradually forms, enclosing its cargo within a protective membrane. The budding vesicle narrows until only a thin membrane connection remains. Within moments, the newly packaged cargo is ready to separate from the Golgi. The vesicle pinches free and enters the surrounding cytoplasm. It now carries its carefully prepared cargo toward its next destination. Our view widens as the secretory vesicle begins its journey away from the Golgi. Although the proteins are fully packaged, they still have one final delivery to make. The vesicle travels along a cytoskeletal track. Motor proteins attach to its surface and transport it efficiently through the crowded interior of the cell. Driven by these molecular motors, the vesicle continues moving with remarkable precision. Cellular transport is highly organized, ensuring that cargo arrives exactly where it is needed. As the vesicle disappears into the distance, the Golgi's role is complete. Our journey now follows this cargo to its final destination, where these proteins will begin carrying out their functions within the living cell.